Okay, guys, we're going to wrap up chapter 7. So when we last met, we had been talking about some environmental factors that influenced microbial growth. We're ready to talk next about relationships, ecological relationships that exist among microorganisms. This is section 7.5 in your book. And we want to begin by understanding that in many instances, there are what are referred to as symbiotic relationships that exist between microbes and other organisms. Symbiotic meaning a very close, rather intimate relationship has evolved between the microbe and the other member of that relationship. And in some situations, there is a necessary, intimately necessary relationship, because without one another, neither of those organisms would survive. And we're going to, again, talk about some of those different examples as you see here introduced on this slide. We can think again of this as being a possible plus-plus relationship whereby the microorganism and the other individual both benefit from that relationship. Sometimes they are obligatory in the sense that without the other, that individual would not be able to survive. Then we have the commensalistic symbiosis, indicated by a +0, meaning that one benefits and the other is really neither harmed nor helped. And then finally, parasitism, which you've all, I think, have been exposed to. You have an understanding of what that refers to, where one individual benefits, the parasite benefits, while the host is negatively impacted by that relationship. So let's give some examples of mutualism, commensalism, and parasitism. And there are many of them out there. One example that is often depicted in many biology textbooks, including yours, is the mutualistic symbiosis that exists between bacteria and plant roots. Now, I'm not talking about mycorrhizae, although one could argue that also is a mutualistic symbiosis. This is a little bit different. We're talking here about certain types of bacteria that live in little nodules that are connected to the roots of plants. And we often talk about legume, the legume family, as those plants that harbor in these little nodules specific types of bacteria that are beneficial to the plant. What the bacteria does is it's able to fix nitrogen fixation, it's called. So what occurs is nitrogen in the environment, in this case in the soil, is basically converted into usable forms that the plant can then make use of directly. And plants need nitrogen. Remember, it's one of the big 6, chopsin. Plants need nitrogen for all organic molecule synthesis. In fact, all cells need nitrogen. The bacteria is benefiting from having a home, so to speak, the nodule of the root, and the plant is also providing adequate moisture and other important compounds that the bacteria is using as it survives and reproduces and grows. Another interesting example is one that we can see if we go to the coral reefs. And I've had a chance to actually see this quite often when we go down to Panama. There's a particular type of jellyfish whose genus is Cassiopeia. This is also the term that you may have heard of before. If you've ever been outside looking at stars at night, there's a constellation called Cassiopeia. But this is a jellyfish, a genus. And inside Cassiopeia, the upside down jellyfish, it's called, and here you see it. In the tentacles of this upside down jellyfish are symbiotic dinoflagellates. Now, we did talk briefly about dinoflagellates in labs about a month or so ago when we surveyed the different kinds of eukaryotic microbes, if you remember. But the reason that Cassiopeia is called the upside down jellyfish is because it orients itself with its bell, which is this part right here, facing downward, and its tentacles face upward. Now that's very different from the normal jellyfish that you've seen maybe in an aquarium, where it's swimming around, with its bell kind of contracting and causing the water to get pushed, such that the jellyfish sort of swims and the tentacles extend down from that. In Cassiopeia's case, it orients itself upside down because these dinoflagellates, some of them are photosynthetic. So they're able to undergo photosynthesis, and some of those byproducts are utilized by the jellyfish in the same way that some of the byproducts of the nitrogenous bases, products rather, produced by the bacteria are beneficial to the plant. So the Cassiopeia organism, the jellyfish, benefits from the photosynthetic products produced by the photosynthetic dinoflagellates. And the dinoflagellates, like the bacteria in the nodules here, have a place to live. The Cassiopeia jellyfish provides adequate nutrients and other things for the dinoflagellates. So they're both relying on one another. Another example that you see in this photograph are corals, which are different than jellyfish. They're related, but corals are sedentary, and they have a hard outer calcareous sort of exoskeleton. But in that exoskeleton are little stinging-celled organisms, somewhat related to starfish, actually. And in the tentacles of these little cnidarians exist similar dinoflagellates. And they're often what contribute to the beautiful colors that we see in various corals, reds and oranges and some green and blue and all sorts of really cool colors. And it's been discovered that as a result of global climate change, and in particular elevation in seawater temperature, is causing corals to get rid of their symbiotic partners, these dinoflagellates that also live in the tentacles of the coral organisms. They actually expel their dinoflagellates, they kick them out. The elevated water temperature causes this phenomenon. And when the corals lose their symbiotic dinoflagellates, they can no longer photosynthesize, and therefore those products of photosynthesis aren't available to the coral. So the coral actually dies, and it undergoes a process called bleaching. So coral bleaching has been a problem that dates back at least 40 years. And in some parts of the world, there are vast amounts of corals that are dying, that you see this just sort of white appearance to the reef. The reef is dead. The coral organisms are dead because they have expelled their symbiotic dinoflagellates. And not to get into a lot of detail here, but coral reefs are sort of the rainforest of the ocean. So many organisms rely on coral reefs that to see these coral reefs become degraded and eventually die has major ramifications ecologically in the oceans. So we're very, very worried about that. Commensalism, this plus zero relationship where one benefits, the other one is neither harmed nor helped. Your book gives an example of a particular interesting mite that lives in and around our hair follicles that metabolizes some of the lipids and other materials that we produce. Doesn't cause us any harm, but the Demodex mite is a very happy mite. Micrococcus luteus, we started to work with that in lab. For those of you that are in labs, you've heard that particular bacterium before. It lives on our skin, doesn't hurt us, doesn't harm us, but we again provide those necessary nutrients for Micrococcus to do quite well. Parasitism, the plus minus relationship, again, whereby the host is negatively impacted, but the parasite gains some benefit from that. We've talked about this quite a bit. The Plasmodium protozoan, remember, harbored in the Anopheles mosquito, the vector here. When the female mosquito bites you to get the blood for its eggs, it can introduce this Plasmodium protozoan into your body, and it goes through, again, a very complex series of life cycle changes, one of which is involving the invasion of red blood cells. And one of the symptoms that we said of malaria was when those red cells lyse. That's one way you can sort of determine what particular species of Plasmodium is causing the malaria. Some cycle every 48 hours, some every 72 hours. And we just finished up the virus chapter in chapter 6. And so obviously, if you think about viruses and their biology, they are intracellular parasites. They have to get into the cell to complete their life cycle, right? So that's another example of a parasitic symbiosis. We could talk about many other examples of mutualism, commensalism, and parasitism. And it doesn't always involve microorganisms either. Well, one particular interesting ecology that is mutualistic involves these insects. I wanted to take a couple minutes and kind of talk about this very fascinating mutualistic symbiosis. We're off topic just a little bit, but I think it's worth taking a trip down to the tropics. Let's go to Costa Rica, where there live different types of ants, lots of different kinds of ants, and they actually rule the tropics. Believe it or not, these little insects have a major ecological role to play. But there's a group of them called leafcutters for obvious reasons. They take their mandibles and they literally trim little pieces of leaf off a plant. And you can see that in the photographs here, where they've literally taken their mandibles and they cut this small little piece of leaf. It's probably just a couple millimeters in size. It's really tiny. And they've got to then carry that back to their nest. And sometimes the defoliation process as a result of the action of these leafcutter ants can be very noticeable, as evidenced by this photograph in the lower half of the slide. And you might think, well, you know, is the plant going to die as a result of this defoliation? And the answer is typically not. There's such a fast reproductive growing process in the tropics with adequate rain and temperature and moisture and such, that a lot of plants are able to generate new foliage almost as quickly as the leaf cutters, you know, cut them up, cut up the leaves. And the ants know not to denude an entire plant. There's so many other plants out there that it's rare that a plant would really suffer any long-term impact by leaf cutters. They just can move along to other plants and don't generally stay on one all the time. So again, here's a photograph of a piece of leaf that this particular ant is carrying. And what I want to point out to you is that in ant biology, there's often a series of castes within the larger community. What I mean by that is certain ant species will have 3 or 4 individuals within that larger nest. There's soldiers, there's workers, there's a whole system of duties that different ants do. Now, they're all the same species, but some are big. The soldiers are really big. The workers are a lot smaller. These little minims that you see, a photograph of this little minim on this piece of twig, they have a fascinating role to play in the leafcutter family. These little minims ride shotgun. Well, what I mean by that is in the old Westerns, you might remember seeing the stagecoach driver up on top, and next to him seated would be a fellow with a shotgun to protect the passengers in the stagecoach from being robbed. So this little guy is sitting shotgun, and what he's doing is he's protecting his brother, the larger larger soldier down below, from possible injury that might occur as a result of a wasp that likes to parasitize the ant. What the wasp would do actually is it lays an egg on the body of this ant, and the egg would hatch, and then it would burrow into the ant and complete its life cycle, thus killing the soldier. So this little minim protects the soldier. If a wasp comes by, it'll try to bite it, preventing it from parasitizing its brother. It's amazing, the intricate biology in the tropics. It's just astounding. This is actually a path through the rainforest. There are so many ants that travel these same paths that they wear down a trail. You can actually see the trail right through, grassy lawns of people's backyards or through the rainforest. And who would ever think that that could happen? But over many, 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 many decades, or maybe even hundreds or thousands of years, these ants have been using these same general trails to the point that they're worn down. It's truly amazing. So once the ants take all of this leaf material, these little pieces of leaves, back to their nest, they take it underground. So these ants have subterranean nests, and they're quite complex structures, very large. And what they then do is they transfer these pieces of leaf and twigs and so forth, maybe even flowers, to other members of the same species that will chew that leaf material up, and then will use it to culture a fungus. Okay, and here you can see in white these little ganglidia, they're called. These are the fungi that the ants basically farm, and that's what they eat. They eat the fungus. They don't eat the leaf material. They're using the leaf material as a medium upon which to grow the fungus underground. And what's absolutely astonishing is the fact that these ants are able to culture this fungus in pure culture. And if you're in lab, you know what I mean by that. Pure culture means there's only one species of fungus being farmed. It's pure. It's a single species of fungus, and that's pretty astounding when you consider surrounding this nest is dirt, and in dirt are hundreds of different species of fungi. So these ants are able to farm in such a way as to maintain this pure culture. They're quite the microbiologists, aren't they? This upper photo is just sort of interesting. It shows the larger queen ant. She does all the laying of the eggs. It's not dissimilar to bees, honeybees, where the queen is the sole reproducing female. She lays tens of thousands of eggs in a year, actually even more than that. And every once in a while, when the colony needs to move to a different area, there will be some cells that are formed here, some of the larvae that will form into other queens, and they are winged. That's what's being shown down here, winged queens, and they'll fly off and begin laying and start up a new colony. Okay. We've talked a little bit about biofilms back in chapter 4. It was introduced anyway at that time. And I want to take some time now for us to delve into the biology of biofilms, because we could take an entire course on biofilms. It's such an interesting topic. And I'm going to ask you to click on this photograph. I'm not going to do it right now, but I'd like you to do that and watch that short little animated video that goes over sort of the biology of biofilms. We'll go over it here as well in a few more minutes, but it's just a nice introduction that I'd like you to click on and watch. And so we're talking about these structures formed by bacteria, although I have heard and read that sometimes eukaryotic cells can get involved as well. Sometimes fungi, sometimes different types of protozoans can be part of biofilms, but we're going to focus our attention mostly on the bacterial biofilms. And so, as the name sort of implies, film, bio, living film, these structures you can find all over the place. We can find them on our teeth, some of which can contribute to dental caries or cavities. We can find them lining endotracheal tubes or catheters that are introduced into the body. Now, when these come out of the plastic sleeve upon insertion, they're sterile, obviously. But what happens over time is biofilms can form on the inner lining, inner surface of this plastic endotracheal tube or the catheter or whatever the container might be. Here we see the result of many years of biofilm formation in a large metal pipe, it looks like. Yeah, doesn't have anything to give us in terms of scale here, but here we've got another similar metal structure, and somebody's holding it obviously with their fingers, so you can get an idea of how big that is. That's got to be at least an inch or two across, probably inch and a half. And look at how narrowed this has become as a result of this biofilm that's formed within it. Amazing. And sometimes these biofilms can contribute to disease. And I'll be referring to another hyperlink YouTube video that I'm going to ask you to watch that talks specifically about the role of biofilms in chronic disease. This should be a real eye-opener for you guys going into nursing or any of the allied health careers. Not enough is talked about, I think, with respect to biofilms and the role they play in disease. It's just something that I think you all need to be acutely aware of, because it's absolutely critical in the treatment of disease. We don't ever hear much about this, so we should hear more about it. Go back into chapter 4, as I've indicated here, and check out those 2-3 pages, and also reference that figure 4.12. So one important part of biofilm biology is this concept of cells, bacterial cells, being able to communicate to one another. And they do that as a result of chemical signaling between like species, but there can also be communication between unlike species of bacteria. So within that biofilm of your enamel of your tooth or in that endotracheal tube might be multiple species of bacteria, okay? And there could be communication, again, between species of the same, or they can increase their vocabulary, if you will, and can communicate with different species of bacteria. And they work together sometimes, oftentimes, and cooperate among themselves to form the biofilm initially, but also to allow that biofilm to function as this diverse community of microorganisms. And they do that coordination and communication once they reach a certain number of cells, and we reach what's referred to as a quorum. And it's through this quorum sensing phenomenon that we have this communication and cooperation, basically. So the term quorum comes from the concept of a minimum number of members of an organization that are able to conduct business of that organization. Now here you see an old photo of the Supreme Court. I tried to get an updated one, but I just wasn't able to find anything. But it's a very elite quorum, obviously, that helps to determine what laws are enforced and what laws might need to be rejected. Okay. Well, with that concept in mind, we can begin to understand a little more about how biofilms work. And so in your book, you've got this process listed or described there in figure 7.14. And I'm not going to go through all of the steps because some of the videos that you're going to be watching reviews this very, very nicely. But in essence, what we have are the following steps. I guess I will go over it because it is kind of important. You have these forms of bacteria that are referred to as planktonic cells. Planktonic, plankton, algae in the ocean kind of analogy. So here are these floating, motile forms of bacteria. They're often flagellated that are initially inoculating a surface of some sort. Okay, so these free swimming cells, they lose their motility, they settle on a substrate of some sort, and they begin to undergo cell division, which we're going to talk more about a little bit later. A point in time will come when they will begin to produce what's referred to as a matrix. And that matrix material, the chemicals in that matrix, are going to help these cells begin to stick together. So you're going to see pili forming, you're going to see this extracellular matrix forming, all of which function to hold the cells together. Initially, they adhere to the substrate, and then they begin adhering to one another. And when they reach that minimum number needed to conduct business, once they reach a quorum, then what we see happen is the cells will begin to secrete chemicals. And these chemicals are sometimes referred to as inducer molecules. So we're showing the chemicals being secreted by the cells here as these blue little dots, little circles. So out come the inducer molecules. And what these inducer molecules do is they induce a change in the gene expression within the cell. Okay, so you have to think about this a little bit. Here's that single circular chromosome that we know all bacteria have. In the presence of the inducer molecule, which was secreted by nearby cells of the same species, oftentimes, or it could be of different species, the inducer molecule gets into the bacterial cell, it associates with the DNA, and the result is the transcription and translation of specific important proteins. So you got to remember what transcription and what translation are. They make use of DNA and RNA to make protein. So in this case, the bacterial cell is producing a quorum-dependent protein. In other words, this protein, which is secreted by the cell, would not be produced unless there had been a quorum reached until a sufficient amount of inducer molecule had been secreted, and the result being adequate amounts of protein being made. What do these proteins do? Well, they can do all sorts of things. The example that this figure has in your book talks about is how some of these quorum-dependent proteins might actually act as enzymes. Remember, all enzymes are proteins. And what enzymes do, remember, is they catalyze reactions, help reactions go more efficiently. Some enzymes are involved in degradation sorts of reactions. Other enzymes are involved in anabolic synthesis types of reactions. This example for giving here is a catabolic process whereby the enzyme is degrading some food particles, and then presumably the end products are then absorbed by the bacterial cell and made use in some positive way by the cells. The cells could never have obtained that digested food unless the enzymes had been there to break the food down. The enzymes would not have been made unless there had been adequate inducer molecules absorbed to synthesize the protein, and we wouldn't have had adequate inducer molecule numbers unless we had adequate numbers of cells, i.e. a quorum reached. So all these things go hand in hand, and they work through in a sort of stepwise fashion. So you need to spend some time and get a good sense of what this biofilm formation involves and what quorum sensing is. It is, again, critical to understand before we can delve into how this may play a role in pathogenicity and in how bacteria can cause illness and chronic disease in you and me. I'm going to, again, ask you to watch this short little video. I've sprinkled several videos in here for you to watch. That, again, talk about how these inducer molecules work. So I'm not going to play that right now, but make sure you watch that.
It would be.
We have here 3 YouTube videos. I want you all to watch the first and second videos. And if you're interested in watching this third one, you're welcome to do so. But these are 2 must-watch videos. They do a fantastic job getting into a little more detail about biofilm biology. This TED Talk is especially interesting. So again, make sure you watch those. You're going to see some stuff on the test on this. Okay, we're going to shift gears a little bit in the next section, 7.6, looking at microbial growth. We can think about this at 2 different levels, as I've indicated on the slide. We can think about how a cell itself grows, and we can expand our thinking and talk about how that in turn influences how the population itself changes or grows in this example. The process whereby bacterial cells divide is mainly through a process referred to as binary fission. It's very easy, basically, to get a sense of what that is. And I'll ask you to watch some videos here coming up that goes through that. But in essence, you start with a vegetative cell. Okay, let's say it's Bacillus subtilis or E. coli or Staph aureus. You pick whatever bacterium you want, the vegetative state of the cell. Assuming proper growing conditions, adequate nutrients, all that good sort of stuff, a time will come when that parent cell needs to grow in size, needs to replicate its DNA in its chromosome, perhaps make some more ribosomes, perhaps make some more peptidoglycan, right? Whatever the resulting daughter cells are going to need, the parent cell has to make. Ultimately, once the DNA is replicated and some additional ribosomes are produced, and adequate new cell wall and cell membrane are synthesized in the parent cell, that parent cell is going to elongate and eventually pinch in 2, and you're going to be left with 2 daughter cells. Do not confuse this with how a eukaryotic cell, like a skin cell, undergoes cell division. In that instance, we talk about interphase leading to the 4 stages of mitosis, which you remember are prophase, metaphase, anaphase, and telophase, followed by cytokinesis, forming the 2 daughter cells. Okay, that's how a eukaryotic cell would generally divide. The result is the formation of 2 genetically identical daughter cells. Bacteria will also divide in a very different way, but the result will be the formation of 2 genetically identical daughter cells. So from that standpoint, they're very, very similar in the end result, but how you get there is very different. So don't confuse the 2 processes. So here's what I was just describing a moment ago. The parent cell, the single circular chromosome shown here, of course, in pink, various other molecules, and we could also think of these as, ribosomes, because we know that's an important organelle that all bacteria have to help synthesize protein. So we have the initial elongation of the parent cell. We have replication of the DNA, thanks to the DNA polymerases and other enzymes that assist in that process. We can see the 2 double-stranded single chromosomes have already been produced. They're affixed to the inner cell membrane, it looks like, of the cell, and we're starting to form a little wall here, a little septum down the middle. And eventually that septum will separate, and of course here are our 2 daughter cells. Now, how fast does it take for a single cell to undergo binary fission or transverse fission to produce the two daughter cells. So we talk about the doubling time or the generation time as defining that time frame. So theoretically, if you started with a single cell, E. coli cell, let's say, and you placed it into a broth, a nutrient broth, and you then incubated that broth at 37 degrees Celsius, then you pulled that tube out of the incubator after 24 hours or 36 hours. We'd have a lot of cells in that tube, wouldn't we? We'd probably see some turbidity having developed in that tube and a cloudiness, right? Initially, it was sterile broth, right? It was clear. Now it's cloudy. That's evidence physically of lots of cells. So the idea here is, again, a single cell undergoing binary fission, forming two, each of those dividing to form two of their own. And so we basically double the population with each generation time. And this gets at what is referred to as exponential growth. So if you were to plot on a graph over time, how does the population change? Where we start with theoretically a single cell, and we follow that for a given time frame, 24 hours, 48 hours, 96 hours, a couple days, we would see, if we could plot, we'd see the numbers initially start off slow, but it would eventually get very, very fast, and we'd get super accelerated growth as more and more cells are being added to the test tube. And that generation time, here we're following only 5 generations, right, in this graph. Pretty soon, we're dealing with some major numbers. And that process of how long does it take for a cell to undergo binary fission, as it mentions here, can range from as short as like 28 minutes, upwards of several days. So there's a wide range depending upon the bacterial species and a lot of the other environmental factors that probably would need to be considered as well. So there is some mathematical basis for calculating the population change over time. We're not going to get into the calculus here, but in essence, this particular formula where we have the total number of cells in the population here, n sub f, is equal to the initial population, number of cells, times 2 to the nth power, with n, 2n being the number of cells in that particular generation. The generation time is denoted by the superscript n. So let me plug some numbers in to give you a sense of exactly what this kind of is meaning. There's an example that your book talks about there on page 217. And it says, let's use an example of Staph aureus to calculate how many cells, n sub f, will be present in an egg salad sandwich after it sits in a warm car for 4 hours. We'll assume that n sub I, the number of cells deposited on the sandwich while it was being prepared, we'll assume that number is 10. So 10 bacteria fell off your skin as you were preparing the egg salad sandwich, fell into the egg salad. We're going to use a generation time of 20 minutes, which is not unrealistic. Certain kinds of bacteria can undergo binary fission in as short as like 28 minutes. So this is a little shorter than that, but you'll get the idea. And so we're going to be following this over a 4-hour time frame, knowing again that each generation time is about 20 minutes in length. So by plugging these numbers into this equation, we would predict that after 4 hours, we'd have close to 41,000 bacteria present in that egg salad sandwich. Again, what do you need to know? You need to know what the generation time is. You need to know how many cells were initially inoculated in that sandwich or in that broth or whatever the case may be. And you need to know how far do I want to follow this process. Again, I'm not asking you to memorize this formula, but just to understand that there is a way of fairly easily calculating this if you know a couple of the parameters. I'm going to ask you to watch this little video. I'm not going to play it now. Note the fact that this will not start until 30 seconds into the video. Don't ask me why. It doesn't. Okay, here's a question I think you ought to be able to answer. It sort of applies to that preceding slide. If E. coli has a doubling time of 20 minutes, and you start with 5 cells, how many will you have after 3 hours? Can you figure that out? You start with 5 cells. After 20 minutes, that becomes 10. After another 20 minutes, that becomes 20. In another 20 minutes, that becomes 40. Okay. How many will you have after 3 hours? Well, the answer is 45. 45. So you have to figure out how many generation times there are and just do the math. So there's actually, what, 9 generation times. So see if you can figure that out. One way that we are going to explore this phenomenon of population growth in lab coming up in about 2 or 3 weeks is by utilizing something called the viable or standard plate count. So let me explain kind of what this is looking at. Let's say that we start off with a sample of sterile broth, liquid broth. And we're going to inoculate that sterile broth containing 500 mils of solution with theoretically a single cell. If we could do that. This particular culture that we're going to be growing in our flask is going to be placed into a 37-degree incubator. And then every 20 minutes, we're going to pull out 1/10 of a mil from the flask every hour, every 60 minutes. We're going to pull out a 0.1 mil volume. And so there's the 0.1 mils, we'll pretend, in this small little bitty vial. And that is going to then be plated onto a sterile Petri plate. That Petri plate is going to be placed into the incubator. And then we will examine it for some CFUs, which are colony forming units. Remember hearing about that in the lab recently. Okay, so that's what's going on here after an hour following inoculation. An hour after that, we're going to pull that flask out of the incubator again. We're going to extract aseptically 0.1 mils. And we're going to put that onto another sterile Petri plate. And we're going to incubate that for 24 hours. And we're going to pull it out and we're going to look for CFUs. In this case, we've got 2. And so every hour we do that. Looks like we're doing this about 10 times, right? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Yeah, 600 minutes is 10 hours. So every hour we We come back to the lab, we pull out 0.1 mils, we place that onto sterile Petri plates that are then incubated, and we come back after 24 hours and we evaluate each of these plates. And look what we find. We see the number of colony forming units increases as time goes on, which makes perfect sense, right? Because as this flask is incubated, those cells are undergoing binary fission. And if this was E. coli, it would be like every 20 to 28 minutes, right? You'd be doubling the population. Now, what they're doing here is they're making an estimate of the number of cells in the flask by multiplying these values by the volume in the flask, basically. So there's a simple mathematical dilution calculation made. So these are CFUs, the number of colony forming units that we count on the plate, and then this is a conversion, an estimate of the number of cells in the flask. Okay. So this is one way to assess or follow populations in an artificial sort of environment in the laboratory over time. If we were to do this and monitor that process beyond 10 hours, let's say we went for 24 hours. Okay, so every hour for a whole day, we sample that flask. Maybe even we go beyond 24 hours. Maybe we go 36 or 48 hours. Every hour we come in, take a sample, plate it, so on and so forth. What would we see if we could graph that entire big experiment? Well, we'd form what's shown or described as a growth curve for that particular species of bacteria. And it would exhibit this particular shape, theoretically. So here we have time 0, that's when we began the experiment. And here we're going 40 hours, or let's say 48, close to what, 2 days, right? What we would find is that initially the population curve, growth curve, would exhibit what's referred to as a lag phase, where there's this period of adjustment that the cells make to that new environment. Now there's active cell division going on, of course, because we can see that there is growth. This is the logarithmic number of cells. So logarithmic means 10 to the second power, 10 to the third power, 10 to the fourth power, 10 to the fifth power, 10 to the sixth power. So rather than using 10, 20, 30, 40, 50, we get into such great numbers here that we have to use log-based y-axes. Okay, so that's what this refers to, 10 to the whatever number power. So 100, 1,000, 10,000, 100,000, a million, 10 million, 100 million, a trillion, and so on. So for the first few hours, in this particular example, first 5 hours, there's very, very slow growth. There's growth, but not super fast. Then in the second part, we get into that exponential growth phase that we talked a little bit about, where the population is really shooting up fast. It's growing exponentially. It's doubling and doubling and doubling. And within, what, less than 15 hours, we're dealing with a heck of a lot of cells, 10 to the 9th power, which just 10 hours ago, we were really low. We were like less than 100 cells. Now we're up to whatever 10 to the 9th power is, a trillion. Yeah, exponential growth, maximum growth. The cells are dividing every 20 minutes, doubling every 20 minutes. Things are going fantastic. Everybody's happy. Suitable growing conditions, suitable nutrients. Everything is full steam ahead, 100% growth. But there will come a time when the growth curve will plateau, the so-called stationary phase, meaning the population is not growing. It's not dying either. So we have as many cells being added as there are cells now starting to die off. Now, why would cells start to die off? Well, if this is a closed system, like our flask was, right? We're not putting new nutrients in. We give them a finite amount at the beginning. There's going to come a time when waste products are going to begin to develop in that flask or in that test tube. In this case, we're showing test tubes. And those waste products are going to begin to take their toll on some of the cells. So some of the cells are going to begin to die off. Now, there's still some active cell division going on. So again, growth equals death. So the stationary phase is maintained for a certain number of hours. And then there will come a point in time where we enter the death phase, more cells dying than are being produced. If this was a endospore forming bacteria, this would be the time to start producing endospores, right? Nutrients are being depleted in an effort to save your species. If you're a bacillus or a clostridium, you're going to form endospores. We've talked about endospores, so you know what that means. Not all bacteria produce endospores. So if you're a non-endospore former, it's not looking good. Eventually, that's going to go down to zero if you were to, wait a week, 2 weeks, follow what goes on in that test tube. Eventually, all the cells would likely die because they'd run out of food, and the toxic waste products would begin to have real deleterious effects on the population to the cells. This is a really important concept with respect to what's the best time to target bacteria from a treatment point of view. Well, it turns out that these bacterial populations tend to be most vulnerable in their exponential growth phase, which of course is this. If you can determine the exponential growth phase time in an infection, which is going to be shortly after inoculation, right, within a reasonable time frame, if you can get a drug administered earlier in the process, those dividing cells are much more vulnerable to the effects of antibiotics, disinfectants, and other agents than they're going to be when you enter the stationary phase. Okay? So target them here. You're going to be more efficient or more effective at limiting their growth than if you wait to the stationary phase. Because you might start to begin to develop, for example, endospore formation and other things that might make the cells a little less resistant, or I'm sorry, a little more resistant, a little less impacted, by the drug. So again, target them early on during that exponential growth phase is your best bet often in limiting the cell division process. Because when cells divide, remember, they're making more cell wall, they're making more cell membrane, there's active metabolism going on in that parent cell as it forms the two daughter cells. If you can disrupt the cellular metabolism pathways, you're not going to let the cell divide. That's the theory. And I think it's more than just theory, it does work. Okay, I want to just introduce an interesting chamber called the chemostat. And here we see a couple chemostats in laboratories. And these bottles look rather small, don't they? Probably a couple liter size volumes for these chemostats. But look at these other guys here. These are much, much bigger. And in some industries, the containers can be enormous. Now, what do these chemostats do? How do they work? Well, remember earlier we talked about how in our example of the 500 ml flask, or with respect to the test tubes in the population growth curve, that those were closed systems, right? Meaning we weren't introducing nutrients, nor were we taking away waste products. It was just a closed system, literally closed off. In a chemostat, we are siphoning off the waste products produced by the cells at the same time we are introducing fresh nutrients. This would allow the population to be maintained at a very high level, wouldn't it? It wouldn't enter the death phase. It might enter the stationary phase possibly, or we can have this population growing at the very steepest part of the curve. Why would we want to do that? Well, if we are Pfizer or some major pharmaceutical company, and we're in the business of making antibiotics, any pharmaceutical company, I guess, should pick on Pfizer. Could be Moderna, could be AstraZeneca. There's a zillion of them. Siemens, right? A lot of them. One way that these companies can crank out copious amounts of antibiotics is to use huge chemostats to promote the growth of these bacteria. And then what they do is they are able to extract antibiotics from those bacteria and basically synthesize and utilize those drugs to the point at which they're then packaged and sold to you and I, or given to us once we get a script from our doctor for a given antibiotic. Most of the antibiotics originated from bacteria or fungi. And even today, while we have some synthetic man-made antibiotics, there's still quite a few that are made by bacteria and certain kinds of fungi that we grow in big industrial chemostats in pharmaceutical fields. So it's really kind of cool. We're going to wrap up the chapter and talk about some other mechanisms or ways in which to quantify populations of bacteria. And this particular slide is talking about a technique that we're going to be using in lab coming up very shortly. We're going to be using the viable plate count method in concert with the use of the spectrophotometer, which is this machine shown here on the slide. We're going to use this concept of how, when a broth is initially inoculated, it's clear. This is a sterile broth. We can see through it, can't we? But when we inoculate that broth and we put it in the incubator for a certain time frame, we pull it out, we're going to see that it's cloudy or turbid. So turbidometry is the process of analyzing the turbidity of a liquid. And that's what this spectrophotometer does. What you basically do is you take a liquid of known turbidity and you place it into this device, and then the machine will shine a light through it, okay? And you'll read off a little dial here. This is a digital, obviously, number, but on some of the Spec 20s that we have, there's going to be a needle with some numbers on it, so you can use either system, either brand or either type of spectrophotometer will work. So if there's no turbidity in the test tube, like we see here on the left-hand side, the light's going to pass through that. There's going to be a very low absorbance value recorded. A very low turbidity, same idea. If you analyze, however, this sample and put it into the machine, when that light is passed through it, yes, some will get through, but there's also going to be a lot of diffraction and refraction of the light because we have cells present. So the absorbance value will be higher due to the presence of cells, as opposed to if there were no cells, you'd have a low absorbance. So the more the cells, the more the turbidity, the higher the absorbance value. So we're going to be doing some work on this, as I said, in a few weeks. So you're going to be able to see this. You're going to be putting some test tubes into the spectrophotometer. Once we calibrate it, we're going to be measuring some absorbance values, and we're going to do some graphing, and we're going to talk about this. Another way that one can enumerate bacteria is by doing a direct cell count. And that's sort of what this slide sort of shows. There are certain types of microscopes, or microscope slides, I should say, called hemocytometers that we use to get an idea of the number of red blood cells or white blood cells in our body. When we get to this in lab, I'm going to show you a hemocytometer, and you'll see once I put that on the microvideo system, you'll be able to see, once magnified, that there's little grids etched on this special glass slide. So if you can dilute a given sample and place that sample onto the slide and look at it under the microscope, you will be able to count the number of cells present in a particular grid pattern. You multiply that by a dilution factor, and you can get some idea of how many cells were in that test tube that the sample was withdrawn from. Okay, so it's the same idea as we had a little bit earlier. Instead of taking a sample out and plating it and counting the number of colony forming units in the viable cell count or standard plate count technique, we could take a sample from the flask and put it on a special slide and count the number of cells present, multiply by a dilution factor, and we can get an estimate of the population in that test tube at that time frame, whenever the sample was withdrawn. So it's a manual process. It's a little time intensive. This mechanism, this process isn't used anymore today because it just takes so darn much time to do. But theoretically, one could do this and get fairly decent data. Today, we use much more automated systems that are extremely accurate. We use flow cytometers, they're called. And this gets into some pretty sophisticated physics and chemistry and biology. But basically what they do is they will take a sample, a bacterial sample. They will subject that sample of cells to certain types of fluorescent dyes. And then they will stream that sample through a very, very small tube within the flow cytometer machine. So here's the sample of cells that have been stained with special sorts of fluorescent dyes. They pass through the flow cytometer. So flow means cells moving through. And there's a laser rather than a light like we have in the the spectrophotometer that we talked about a moment ago. This is a laser. It's shining through. There's detectors, a detector on the opposite end of the cytometer. And depending upon the charge that is present on the surface of the cells, don't ask me how they do this, but somehow they're able to subject the sample to electromagnets, and then the positively charged cells fall in one region, the negatively charged cells fall into another region, and the eliminated cells, I'm not sure exactly what that refers to. Not sure what eliminated means. But when you graph the data, and here we're looking at two different species of bacteria, obviously Staph aureus and E. coli, what they're able to determine based on the color that fluoresces. Remember I said that we're subjecting these different color fluorescent dyes. So if you get green fluorescence or you get red fluorescence, will determine whether you get staph if it's green or E. coli if it's red. And not only that, but they are able somehow to determine how many live cells pass through and how many dead cells pass through. That's pretty crazy. They use similar flow cytometers, as I've indicated here, to do blood counts. Remember earlier I talked about the hemocytometer that used to be used in the olden days to determine RBC and white blood cell counts. Today, they use flow cytometers to determine red blood cell count, white blood cell count. For all I know, they may even be able to do differential white cell counts this way too. In other words, differentiate, lymphocytes from monocytes, from eosinophils, from basophils, from neutrophils. I mean, it's just astounding. Being able to even analyze samples for cancer, they can detect cancer cells from non-cancer cells. They can differentiate gram-positive from gram-negative cells. Like they're doing here. This is gram-positive, this is gram-negative. It's amazing, the technology. These flow cytometers are astounding pieces of instrument that we use each and every day in laboratories. Gone are the days of the manual evaluation of blood counts and all that stuff. It's all automated today. Okay, that I think wraps up chapter 7. Yes, it does. So I hope you are getting into chapter 7. Let's try to wrap that up soon. And we'll leave it at that.